My name is Jeff Martin. I'm here with Kevin Flynn from CaseBioscience. We're out in Colorado. Kevin, what are you guys doing over at Case?
So we design cryopreservation solutions for cell therapies. We base this off of key learnings in assisted reproductive technology. So our company was really founded on merging assisted reproductive technology and cell and gene therapy and cross-pollinating the ideas and expertise from both of these fields. So, but in terms of cell therapies, we focus on the solutions themselves for cryopreservation to try to improve them and provide diverse.
So should I know what assisted reproductive technologies are? What is
assisted reproductive
ART,
technology?
Yeah, what is
so
that?
This is what you commonly think of as IVF, in vitro fertilization,
Okay.
as well as the embryo culture that occurs afterwards,
Okay,
and
got
then cryopreservation. So it's these steps that have been in process, that have been worked on for decades now, and there's
I see,
a mature regulatory landscape there.
Yeah.
So we've taken that, and our founders came from that field and thought that they could apply those learnings
Why
to
has
cell
nobody
therapy.
else done that yet? That sounds like a very common or
It's a great question, because if you think about it, assisted reproductive technology is the original cell therapy, because
Yeah.
you're taking cells out of the patients, you're doing something in vitro or ex vivo, doing some manipulations, and then you're putting them back into the patient.
Mm-hmm.
So it's really the original cell therapy, which
Yeah.
was in the late 1970s.
It's that old?
Yeah. So
All
Louise Brown is now a little bit older than me. I think she's 48. So
she was the first baby.
She was the first IVF baby.
Nice.
Yeah.
Nice. And you're so at Case, your big thing is cryopreservation. You're like the experts of cryopreservation.
Yeah, so we had a couple of our chief scientists came from the ART field, and they came from the area of cryopreservation as well, and where they specialize particularly in DMSO-free formulations,
Yeah.
which are important for many reasons. One is to mitigate toxicity
Toxicities.
and things of that nature, and they use different technologies, vitrification, that could have applications in cell therapy as well.
See, now I come from a research lab. I'm a immunologist by training. When I hear DMSO-free cell freezing, I'm thinking you're going to get cell death.
Yes.
So not to get proprietary, but so DMSO really is meant to kind of like protect the cells from the crystallization of the media,
Right.
and like not rupture
From
the cell
ice
membrane.
crystals, yeah.
There must be some kind of other solution you guys use. It's non-toxic. Is
Right.
What I'm going to kind of. I don't want to get into
Right. Yeah. So don't get me wrong.
information.
DMSO has been fundamental to the expansion and the foundation of cell therapies. You know, it's the reason why we're here, but it's not the best solution for all cell therapies.
Yeah.
Like you said, it can be quite toxic, and for example, in toxicity assays, like if you do a MTT assay or Alamar
Right.
Blue assay, it's the positive control you use to
You can
kill
your
cells.
cell with DMSO.
So there are other solutions, DMSO-free solutions, that have a better cytotoxicity profile. They're potentially safer for patients. Things like sugar alcohols and glycerol are commonly used, and they act in a similar way as DMSO, hydrogen bonding, to try to prevent that ice crystallization.
Yeah.
They're a little trickier to work with, but if you design your formulations in the right way, they can be just as effective at protecting cells from the cryo injury.
Crystallization.
Yeah.
So the standard method with DMSO-based freezing is just thaw, spin down really quickly to get them out of the DMSO.
Yes.
And you're saying that maybe immediate post thaw viability is like the gold standard of cryopreservation, but that might not be the best way to look at it.
Right. No, immediate. I think people realize that looking at immediate viability is not a good way to judge your product. You may get 95% viability and 80% recovery right out of thaw, but
Right.
if you look a day or 2 days later, that recovery has dropped to 50% or even lower. In the case of T cells, you may see a dramatic drop in the number of functional cells. So what we do at CaseBioscience is we always look at the long-term viability, the 3 days, 4 days, 5 days. Are the cells proliferating? Are they functioning the way you expect? Can they kill their target cells? Things of this nature. And so you have to go beyond viability. And this also is in terms of what you expect out of your solutions from a quality standard. You need to have consistency, scalability, and clinical readiness, I think, as well.
Sure. So this is a dumb question probably, but when it comes to the differences between allogeneic and autologous cell therapies, is there more of a risk in the freeze-thaw process for allogeneic than there is for autologous?
I think so. I think
Okay.
you hit the nail on the head, because a lot of the people we talk to that are doing allogeneic therapies, you're filling hundreds of vials or
Right.
thousands even, or hundreds of bags, and this takes time, and you're sitting at potentially at room temperature for multiple hours.
Yeah.
And when you
That's not good.
it's not good when you're dealing with a DMSO solution.
Yeah.
So that's where non-DMSO solutions would shine, in that
Interesting.
benchtop toxicity is reduced, and I think it makes more sense for certain processes, especially ones that are time consuming,
Yeah.
to switch to DMSO free solutions.
So this is very interesting because one of the major problems with allogeneic T-cell therapies has been the lack of persistence in the patient's body. And the lowest hanging fruit reason for that is just rejection. Like, oh, we didn't stealth them enough,
Yep.
and they're getting rejected.
Yes.
So the big push in allogeneic cell therapies is in like molecular modifications to make the cells more stealthy,
yes.
right? But what I hear you saying is maybe it's actually simpler than that. Maybe it has to do with the cryopreservation of the cells.
I think it's definitely a key factor. I mean, of course, you do have the issue with allogeneic of evading the immune system, but if you don't know that your product is surviving long term, like I said, if you're getting the 90% recovery right out of the bat, and that's what
And you just make
you're
the
judging,
assumption.
you make assumption that
Yeah.
they're all going to live. That's a bad way to proceed.
Right.
So
Right. So you guys come from the ART IVF field.
yes.
How is your experience with like knowing how to go about highly regulated ART workflows? How does that change or influence your expectations for cryopreservation in cell therapies for like cancer?
So, yeah, coming from a more regulated field like ART, well, we think, first of all, we think ART sits at a regulatory sweet spot that cell therapies have failed to define. And so if you look at what happens in ART, the materials are used for IVF, for embryo culture, and for cryopreservation. So you're dealing with materials that are in contact with human cells.
Yeah.
They're not intended as drugs, but yet they require more complex regulatory standards, specific guidelines for traceability and risk management as well.
Well, and viability would be a massive concern with IVF.
Oh,
I mean,
yes,
like you're talking about
of
a
course.
human being
Yes.
versus,
A couple
a population
cells.
of T cells.
Yeah, exactly.
Which is much, we don't really care if we get 1 million T cells that die within a day. We still have, a lot more. I didn't think of that. With IVF, it's like we have a dozen shots at this, maybe, right?
Yeah, I mean, in a cycle at IVF, you may have 20 to
Right.
40 oocytes, potentially, a dozen good blastocysts.
Right.
And so that's huge.
Yeah,
If you
that's a
lose
big
one
deal.
of them, it's a huge deal, because it's a
Right.
potential human being.
Right.
And that's why, I mean, not everything directly translates, because these vitrification procedures occur in small volumes and are a little bit different. But you can, and we have made strides to learn from those procedures, especially in the formulations. The slow cooling or controlled rate freezing is still kind of the standard in cell therapies, but there's room for developing new ways for freezing cells, or actually just biopreservation in general.
Sure,
Doing maybe avoid the cold chain altogether in certain
That would be huge.
circumstances. Yeah.
That'd be huge. So who do you guys primarily work with? Are you guys working with CDMOs? Do you partner with the therapeutic developers themselves? Who is your major, partner?
So our major partners are, we partner with CDMOs, but also the early stage therapeutic companies, because we want them to see the benefit early on,
For sure.
before they go into clinical trials. But now we're hoping to partner more with more mature cell therapy companies. We just had our DMF filed, so
Nice.
we have a reference for them to go to the FDA with the INDs. So yeah,
Do you have any clinical programs right now, or do you not want to?
So we can't really say exactly what's going on, but definitely a lot of testing, and people are using our stuff, and we're getting good feedback, and it's a wait and see approach here.
Yeah, it's one of those things that I wouldn't have thought about that when I think of cell therapies. cryopreservation is not the top of the of the food chain,
and that's unfortunate.
Right?
It's often an afterthought,
Right.
and it can be detrimental to your cell therapy product if you don't do it right from the beginning. You have to think about it
That's the thing
early
too.
on
you
in development.
Change your process halfway through.
Yeah, exactly. Yeah, that could be
interesting. So the idea that allogeneic cell therapies are being limited by the cryopreservation mechanism is a completely new thing for me, and probably for a lot of people too, I would imagine.
Yeah.
That would be a really
we have
huge revelation. If that was the case.
One partner that has been extremely concerned about this, like like I said, filling multiple vials over multiple hours and seeing the drop in cell viability, and then you freeze these vials and then you come back out and so in some cases they do direct injections out of the vials, and your product is 50% viable. It's not a good thing to go to the clinic with.
And it's probably easier to blame something else
at
that point.
Right. You don't know what the causal issue is. But I think people are catching on, and seasoned cell therapy manufacturers, I think, realize how important cryopreservation is and biopreservation in general. So I think we're on the right track to getting a little bit better at the processing of cells and realizing how important cryopreservation is.
Yeah. So one last question then. So you're talking about cell therapy, like therapeutics, but is there any other pillar that you're kind of thinking about this could be useful in, or is it just mainly
So it's useful in anything that you have to freeze cells or biologics in general. So
even like academia, could they benefit from this?
Oh yeah, for sure. I mean, academic programs, absolutely, and especially if you want consistency in what you do. I mean, we do everything according to ISO 13485 standards. So this is a regulatory standard.
Yes, I know what that is.
That requires traceability. You need a robust quality management system. You have risk analysis. It's all about consistency, scalability, and quality. And if you apply that to your research, you're going to get more consistent results. And but even more important is for cell therapies. You need that consistency, that quality in what you do, and that's what we bring to the table. Is that we take that manufacturing, that higher level of clinical grade, medical grade manufacturing to cell and gene therapy.
Yeah.
And it's extremely important, and I think also for research. If you want reproducibility, you gotta trust your suppliers and know that having an FDA registered clinical grade manufacturer is a good starting point.
Yeah.